Usually when we talk about a medical diagnosis, there's this expectation of precision.
Oh, absolutely.
Like it feels almost like engineering, right? You break your arm, the X-ray machine hums, it prints out a sheet with that stark, jagged white line.
Right, and the doctor just points at it and says, there it is.
Exactly. It's entirely binary. Broken or not broken. Safe or unsafe.
We are deeply comforted by that kind of clean categorization. I mean, we want the natural world to be visible and, well, easily sorted into neat little boxes.
And then you step into the world of forest ecology and mycology.
And suddenly that X-ray machine is completely broken.
Yeah. We're looking at a diagnostic landscape that is, well, honestly, it's incredibly murky. You think you're looking at a simple brown mushroom on a log, and you just assume it has a simple life.
Right, a simple function.
But that feeling, that exact feeling of looking at something you think you completely understand, only to realize the categories are just shifting under your feet.
Yeah.
That's exactly what we're getting into today.
It really is.
So, welcome to a brand new deep dive, and today we have a very specific mission for you. Our mission is to unpack a single, highly deceptive, and honestly, utterly fascinating organism. We're turning our magnifying glasses onto Cortinarius armillatus.
Commonly known as the red-banded cort, or the bracelet Cortinarius.
Right.
It is an absolutely remarkable subject. And to really get to the bottom of the mystery surrounding this organism, we're drawing from an incredibly thick stack of scientific literature for you today.
The huge stack.
We're looking at comprehensive monographs on the Cortinarius genus as a whole, and we'll be diving deep into a groundbreaking 2011 taxonomic study. This is by Niskanen and her colleagues from the journal Mycology.
Which quite literally rewrote the rule book, right?
Entirely. It changed how we view this entire species. We also have a crucial 2016 toxicological report by Xiao and a team of researchers, which was published in Toxicon.
Good light reading.
Right, exactly. And finally, we're even pulling from ethnobotanical guides that focus on the ancient art of natural mushroom dyeing.
It sounds like a really wild mix of sources.
It does, yeah.
But it makes perfect sense because this mushroom is basically a walking paradox. I mean, it's an organism that is simultaneously a vital architect of the underground internet that keeps our northern forests alive.
Which is fascinating on its own.
Right, but it's also a master of disguise that has actively fooled professional mycologists for over a century. It's a carrier of a terrifying delayed-action kidney toxin, and somehow, despite all of that, a highly prized tool for textile artists.
It's the perfect example of how the natural world just, refuses to be just one thing.
Yeah.
When you look at the red-banded cort, you're forced to completely abandon the idea that an organism in the forest has a single, simple purpose. Every layer you peel back reveals a new mechanism or a new chemical secret or a new evolutionary trick.
Okay, let's unpack this, because before we can get into the invisible ecological web or the chemical warfare happening inside the cellular walls of this fungus, we need to know what we're looking at.
Right, the visuals.
Yeah, we need to build a really clear mental image for you, the listener. Because if you're walking through the woods this autumn, you need to know how to actually spot the bracelet Cortinarius. So what are the physical traits that give this mushroom its identity?
Well, to start building that image, you first have to appreciate the sheer, just mind-boggling scale of the biological family it belongs to.
Okay.
The genus Cortinarius is the largest genus of agaric mushrooms in the entire world.
Wait, the largest in the world?
Yeah. We are talking about over 2,000 recognized species globally.
2,000, what?
It is so massive and so complex that mycologists have had to break it down into various subgenera and sections just to, well, keep their field guides organized.
I can imagine. So where does ours fit in?
Cortinarius armillatus belongs to the subgenus Telemonia, and this particular subgenus is kind of notorious in the scientific community for being the most species-rich and taxonomically frustrating group to study.
Like the problem child of the mushroom world.
Essentially, yes. And within that subgenus, our mushroom is the type species for the section Armillatus.
Okay, so it's essentially the poster child, the flagship mushroom for its specific section.
Precisely.
So if we're just walking through the woods, relying on the naked eye experience, like what you see when you're crouching down on the forest floor, what does this flagship actually look like?
It's a relatively robust, substantial mushroom. The cap is usually somewhere between, say, 4 to 15 centimeters in diameter.
So a decent size.
Yeah. When it's young, pushing up through the soil, the cap is what botanists call campanulate, which really simply means it's bell-shaped.
Okay, bell-shaped, got it.
But as the organism ages and matures, that cap gradually flattens out, It sometimes retains a slight bump in the center, which is known as an umbo.
Right. And what about the color?
The color palette is quite striking. You're looking for very warm, earthy tones. It features rusty browns transitioning into yellowish browns and often a deep brick red.
Oh, nice.
And that red is especially concentrated right at the center of the cap. And the surface of the cap is dry, but it has a specific texture we call slightly hygrophanous.
Oh, I love that word, hygrophanous.
It's a great word.
That means the mushroom actually changes color depending on how much moisture it's absorbed, right?
Exactly.
So if you, as a hiker, find this mushroom the morning after a massive rainstorm, it might look like a dark, saturated brick red.
Yep.
But if you find the exact same mushroom after a week of dry sunshine, it might have faded to a pale, dusty tan.
That is precisely it. The cells expand and hold water, and that changes how light reflects off the surface.
That's wild.
But to truly identify any mushroom in the Cortinarius family, you have to look underneath the cap.
Right, where the action happens.
Right. The defining feature of this entire massive genus, the reason they're commonly called webcaps, is a structure called the Cortina.
The Cortina. It sounds like a stage curtain.
And that's actually a very apt way to think about it. When these mushrooms are young, their delicate gills need protection from insects and the elements while the spores are developing.
Makes sense.
So they're covered by this incredible cobweb-like partial veil. If you look closely at a young specimen, it literally looks like a fine spider web stretching from the outer edge of the cap all the way into the stem.
Oh, wow.
Yeah, and as the mushroom grows and the cap expands outward like an umbrella, opening up that fragile web-like curtain is stretched until it finally breaks.
And when it breaks, it doesn't just disappear, right?
No, it doesn't.
The remnants of that cobweb collapse and stick against the upper part of the stem. It leaves this faint, fibrillous ring zone. It always reminds me of a high-tide waterline on a wooden pier.
Oh, that's a good comparison.
Just a faint, rusty mark, showing where the veil used to attach. But despite the Cortina giving the whole genus its name, it isn't the feature that gives Cortinarius armillatus its specific individual identity, is it?
No, not at all. The specific identity comes from its jewelry.
It's jewelry.
Yeah, the specific epithet, armillatus, is derived directly from the Latin word for bracelet or armring.
Oh.
If you look at the stem of this mushroom, which is thick, robust, usually 7 to 15 centimeters tall, with a distinctly swollen or bulbous base, you will see the dead giveaway.
Which is?
The stem is adorned with 1 to 5 vivid concentric bands of color. They are a striking, saturated orange-red or cinnabar red.
They literally look like little red bracelets stacked up the stem.
Exactly.
But where do those bracelets come from if the spiderweb Cortina only leaves a faint rusty mark?
Right, so those vibrant bands are the remnants of a completely different structure called the universal veil.
Universal veil.
Unlike the partial veil, the cortina that just protects the gills, the universal veil is a thick, protective, egg-like tissue that encapsulates the entire baby mushroom when it is first forming underground.
So imagine the mushroom growing inside a tight red sleeping bag.
That's exactly it. As the mushroom pushes up through the soil and rapidly expands, it bursts out of that universal veil. Because the tissue is attached to the stem as the stem elongates, that red tissue gets stretched, torn, and left behind as these vibrant stacked bracelets on the mature stalk.
That is such a vivid visual. You have this brown, bell-shaped cap, the cobweb remnants, and these bright red bracelets.
It's very distinct.
And then we have to talk about the gills and the spores themselves, because that is the whole biological purpose of the mushroom.
Indeed. I mean, the mushroom you see above ground is just the fruiting body. It's the apple on the tree. Its sole purpose is reproduction.
Right.
The gills on the underside of the cap have a perfect symmetrical arrangement. When the mushroom is very young, the gills are quite pale, perhaps a dirty yellowish or pale cinnamon color.
But that changes.
Drastically.
Yeah.
As the mushroom reaches sexual maturity, it begins producing spores on a massive, almost incomprehensible scale. We are talking millions upon millions of microscopic spores. And the spores of the Cortinarius family have a very specific color. They are a deep, dark, rusty brown.
So as those millions of spores mature and stack up on the edges of the gills, waiting to be caught by the wind, the gills themselves become stained.
Yes.
They turn from pale yellow into this dark, rusty cinnamon color.
That rusty spore print is a non-negotiable feature for identifying a Cortinarius in the field. But here is where the science gets rigorous.
Okay.
To truly separate armillatus from the other 1,999 species of webcaps, mycologists cannot rely on the naked eye. They have to slice a tiny piece of that gill, put it on a glass slide, and take it down to the microscopic level.
Under the microscope.
Under a high-powered lens, the spores of the true red-banded court reveal their architecture. They are ellipsoid, which means they are shaped like tiny ovals, measuring roughly 9 to 12 micrometers long by 5 to 7 micrometers wide.
So tiny.
Incredibly small. And they're not smooth. They have relatively thick walls covered in fine, textured warts, which we biologically refer to as verrucae.
And the researchers don't just look at the shape, right? They perform chemical tests right there on the glass slide.
They do, yes.
One of the sources mentioned Meltzer's reagent. What exactly is that doing to the spores?
So Meltzer's reagent is essentially a specialized iodine solution. It's a classic foundational chemical test in mycology.
Okay.
Scientists use it to test for the presence of certain complex carbohydrates, like starches or dextrins, in the cellular walls of the spores.
And how does our mushroom react?
When you expose the thick, warty spores of armillatus to Meltzer's iodine, they are strongly dextrinoid.
Dextrinoid.
Meaning the structural polymers in the spore walls react chemically with the iodine, causing the spores to instantly turn a dark, rich, reddish brown.
Interesting.
If you apply the iodine and the spores do not change color, they remain what we call indextrinoid, you immediately know you are looking at a completely different species of mushroom, regardless of what the red bracelets look like.
Oh, wow. They also look at the structure of the cap itself, right? The skin of the mushroom?
Yes, the cellular architecture provides further fingerprints. If you look at the gills under the microscope, you will notice a complete lack of cystidia.
Cystidia.
Cystidia are specialized sterile cells that often stick out dramatically from the gills of other mushroom species.
What do?
They sometimes act as microscopic spacers to keep the gills apart, or they hold specialized chemicals. But in armillatus, they are entirely absent.
Completely gone.
Completely. Furthermore, the skin of the cap, known as the pili-pellis, is structured as a cutis. This means the microscopic Hafal threads that make up the skin run flat and parallel to the surface of the cap, rather than standing up straight on end like a microscopic carpet.
Okay, I have to jump in here with an analogy, because you're painting a very specific picture.
Sure, go ahead.
You're basically describing a suspect in a police lineup.
Okay.
And this suspect is wearing very specific, undeniable jewelry. We're talking about bright, neon orange-red bracelets stacked up their arm.
Right.
Now, if I'm a detective, or in this case a forager, and I'm looking for the suspect with the bright red bracelets, why do foragers and scientists still constantly mix it up with other mushrooms? Isn't that an evolutionary dead giveaway? It seems like it would be physically impossible to mistake this mushroom for anything else in the forest.
What's fascinating here is that the natural world is rarely that pristine. And it certainly doesn't care about our field guides.
That's fair.
Those visual cues, as striking and undeniable as they are in a botanical illustration or a perfectly lit photograph, they're highly subjective and incredibly fragile in reality.
Fragile how?
Well, those beautiful red bracelets. They're not made of plastic. They are delicate, microscopic remnants of fungal tissue. A couple of days of heavy autumn rain or just the natural weathering and stretching of an older mushroom can wash those bands away completely.
Oh, wow.
The bright, vivid cinnabar red can quickly fade to a muddy, nondescript, dull brown.
Ah, I see. So the suspect simply takes off the jewelry before stepping into the police lineup.
That is an excellent way to put it. The suspect washes off the evidence. And remember the hygrophanous cap.
Right, where it changes color based on moisture.
It changes color completely based on the ambient humidity of the forest. So relying purely on macroscopic features, the things you can see with the naked eye when dealing with the Cortinarius family, is an absolute trap. It has lured mycologists and foragers into a false sense of security for generations. And that fragility of identification perfectly sets up the ecological and genetic complexities we're about to dive into.
Because to truly know this mushroom, you can't just look at the fruiting body above the dirt. You have to look at what it's physically attached to beneath the soil.
Exactly.
Which brings us to we know what it looks like, or at least what it wants to look like before the rain washes its makeup off. But where do you actually find it?
Right.
Because if you're walking through the woods, the red-banded court doesn't just pop up randomly on a manicured suburban lawn or sprout out of a decaying fallen log. Its location is strictly dictated by an ancient, invisible biological contract.
Yes, the symbiotic contract. This is where the biology becomes truly elegant. Cortinarius armillatus is what we call an obligate ectomycorrhizal fungus.
Let's break that down for the listener, because those are pretty heavy scientific terms. Obligate means it has absolutely no choice in the matter. It has to do this to survive.
Correct.
It never grows from dead wood. It doesn't decay old leaves like a standard button mushroom does. And ectomycorrhizal breaks down into 3 root words. Ecto meaning outside, myco meaning fungus, and rhiza meaning root.
Spot on.
It must connect to the living roots of a host tree. It forms a physical, microscopic sheath over the root tips of the tree, but it doesn't actually penetrate the tree's internal root cells.
Right, that external sheath is called a mantle. And from that mantle, the fungus pushes an intricate web of hyphae between the root cells of the tree, creating a structure known as the Hardig net.
The Hardig net.
This net is the physical interface where the trade happens, and the choice of partner for this trade is incredibly specific. For the true Cortinarius armillatus, the exclusive host is the birch tree, genus Betula.
Birch trees.
In North America, you will see it heavily, almost exclusively, associated with the paper birch. In some temperate zones in Europe, it has been known to associate with hornbeam trees, but the birch tree is its primary ancient evolutionary partner.
This nutrient exchange is like a subterranean stock market. It's a very strict trade agreement. Let's look at the birch tree situation for a second.
Okay.
Birch trees often grow in these highly acidic, nutrient-poor podzolic soils. We're talking about a soil pH of 3.5 to 4.5.
Very acidic.
Now, I'm no botanist, but a pH of 3.5 is incredibly acidic. That's creeping toward the acidity of tomato juice or even vinegar. How on earth does a tree survive, let alone extract nutrients, in soil that acidic?
Well, on its own, the tree would struggle immensely. The low pH locks up essential minerals, making them biologically unavailable to the thick, cumbersome roots of the birch.
So the tree is starving.
Basically. So the fungus steps in. The mycelium of the mushroom, which is that vast, invisible, thread-like network spreading for meters underground, acts as a massive, highly efficient, extended root system for the tree.
It's like outsourcing the work.
Exactly. These fungal threads are microscopically thin, allowing them to penetrate soil pores that a tree root could never reach. Furthermore, the fungus secretes powerful enzymes and organic acids that literally mine the soil.
Mine the soil.
Yeah, they dissolve tiny particles of rock and organic matter to scavenge trapped phosphorus and nitrogen.
That's amazing. It is chemically digesting the forest floor to extract the heavy minerals.
It really is.
And once it mines that phosphorus, it channels it back through the network into that Hardig net we talked about and hands those vital minerals over to the birch tree.
Right.
But the fungus isn't doing this out of charity. The fungus cannot photosynthesize. It has no chlorophyll. It cannot make its own food from the sun.
Exactly. The fungus is entirely dependent on the tree for carbon. So in return for the mined phosphorus and nitrogen, the birch tree pays the fungus with complex sugars and amino acids that it has produced from sunlight high up in the forest canopy.
A perfect trade.
The tree shuttles these sugars down the trunk, into the roots, and across the Hardig net to feed the fungus. It is a perfect, mutually beneficial biological loop. Millions of years of coevolution have locked them into this dependency.
And because of this contract, wherever the birch goes, the fungus follows.
Wow.
This creates a circumboreal distribution. You find this exact pairing spanning across the entire northern hemisphere.
Everywhere the birch grows.
From the birch forests of North America, stretching across northern Europe, all the way into Asia. From the hemiboreal zones where the deciduous forests meet the conifers, right up to the freezing subarctic tundra.
Yep.
If there is a northern birch forest, the red-banded cord is there, acting as a crucial functioning node in this massive underground internet.
That concept, the underground internet, is scientifically referred to as the common mycorrhizal network, and it's vital to understanding modern forest ecology.
Because it's not just a one-to-one connection.
Yeah, no, not at all. These ectomycorrhizal fungi do not just connect to one single tree and stop. Their mycelial threads spread out and connect multiple trees together, often intertwining with the networks of other fungal species. They create a vast subterranean web.
And it isn't just a static web. It's an active highway for resources and information.
Precisely. Through this network, the fungi facilitate the transfer of carbon and water between completely different trees.
Wait, different trees?
Yeah. An old-growth mother tree, towering in the canopy and soaking up massive amounts of sunlight, can actually shuttle excess carbon down into the fungal network.
Now where does it go?
The network then routes those sugars to young, shaded saplings on the forest floor that might not be getting enough sunlight to survive on their own.
That is mind-blowing.
And beyond nutrients, the network transmits chemical warning signals.
Warning signals, like what?
Well, if a tree is attacked by aphids, it can send stress chemicals through the fungal network to neighboring trees. This prompts them to ramp up their own chemical defenses before the insects even arrive.
It's literally an internet.
By connecting the forest in this way, Cortinarius armillatus and its kin significantly boost the overall resilience of the entire ecosystem against stress, drought, and disease.
Okay, let me play devil's advocate for a second, because if you're hiking and trying to identify this, the real world can be confusing.
Of course.
Let's say I'm out in the woods, and I'm standing in what looks to me like a pure 100% pine forest.
Okay.
There are towering evergreen conifers everywhere. And I look down at my boots, and I see a classic red-banded cort with the perfect orange bracelets.
Oh, I see where you're going with this.
Is the mushroom breaking its own biological contract, or am I missing something?
You are definitely missing something. The fungus is never wrong.
Never wrong.
Never. If a true Cortinarius armillatus is fruiting above the soil, there absolutely must be a birch associate nearby. It is non-negotiable biology.
Even if I don't see a birch tree?
Even then, you might be looking up at a dense canopy of towering pines, but if you stop and look closely at the underbrush, you will likely find a tiny 3-foot-tall birch sapling struggling in the shade.
Oh, wow.
Or perhaps the decaying stump of a birch tree that still has a few living roots beneath the surface. That tiny hidden sapling is feeding the fungus, and the fungus is likely routing nutrients to keep that sapling alive.
That is incredible.
Yep.
The mushroom is essentially telling you a secret about the landscape that you couldn't otherwise see.
It reveals how fungi act as hyper-accurate bioindicators. They map out the true living composition of a forest's root system. They expose the subterranean reality.
So if the mushroom is there, the biological contract is active regardless of what the dominant trees appear to be.
Exactly.
So given that this mushroom is a master of disguise, prone to losing its physical identifiers but strictly bound by this elegant, invisible contract with birch trees, you would think scientists had it all figured out.
You would think so.
But this is where the plot thickens considerably. For over a century, since the pioneering mycologist Elias Magnus Fries first named and cataloged this mushroom back in 1838, scientists thought they understood these rules.
Right.
They thought they were looking at one widespread, slightly variable mushroom. But as modern molecular tools evolved, they realized they had been completely fooled.
We are entering the era of the taxonomic shakeup.
I love a good taxonomic shake.
It's quite dramatic. By the early 2000s, genetic sequencing technology had become accessible enough to begin rewriting the established field guides.
Because before that, they just relied on looking at them.
Right. For over a century, mycology relied almost entirely on morphology, what the mushroom looked like, what the spores looked like under a microscope. But morphology can be deceptive, as we've established.
Very deceptive.
The pivotal moment for our red-banded cort arrived in 2011 with a massive, comprehensive phylogenetic study led by Tuula Niskanen and her colleagues.
And they didn't just look at a handful of mushrooms from a local park. They undertook a monumental task. They revised over 1,000 individual specimens collected primarily from across Northern Europe and North America.
A massive sample size.
Yeah. And to understand how these 1,000 mushrooms were related, they didn't just look at the red bracelets. They used a very specific genetic tool, the ITS barcoding region.
Yes.
Let's explain what that is, because it sounds a bit like science fiction.
The internal transcribed spacer, or the ITS region, is effectively the universal genetic barcode for fungi. To understand how it works, you have to look at the ribosomes inside the fungal cells, the factories that build proteins.
Okay, the protein factories.
The DNA that codes for these ribosomes is highly conserved, meaning it changes very little over millions of years. Because if it mutates too much, the cell dies.
Right, don't mess with the essential machinery.
Exactly. However, between these essential coding regions, there are spacer sections of DNA, the ITS regions. These spacers don't code for critical proteins, so they are free to mutate and evolve at a much faster rate.
Okay, so if I can offer an analogy here.
Please do.
Think of the ITS region like the ISBN barcode on the back of a book.
I like that.
You could have 1,000 books that all have the exact same brown leather cover, the exact same dimensions, and the exact same red ribbon bookmark. to Elias Magnus Fries in 1838, looking at the covers, those are all the exact same book.
Right.
But if you take a scanner and read the ISBN barcode on the back, you suddenly realize that one is a romance novel, one is a calculus textbook, and one is a spy thriller. The barcode reveals the true identity hidden beneath the identical covers.
That is a perfect analogy. By extracting the DNA from these 1,000 dried mushroom specimens using a process called polymerase chain reaction, or PCR, to amplify that specific ITS space or region, and then running it through a sequencer, the Scannon's team could read the barcodes.
And read the barcodes they did.
They mapped out the exact evolutionary family tree, the phylogeny. They then cross-referenced those genetic barcodes with the physical characteristics and the ecological data, specifically noting which types of trees the specimens were growing under.
And the result was an absolute shock to the taxonomic system. The traditional broad concept of Cortinarius armillatus, the one that had been in every field guide for a century, wasn't one mushroom at all.
No, it wasn't.
It was a complex family of imposters. They discovered sibling species that were almost visually identical to the naked eye. But genetically and ecologically, they were completely distinct entities that had emerged millions of years ago.
They essentially split the atom of the red-banded cort. The new taxonomic reality determined that the true Cortinarius armillatus, the genuine article with the bright orange-red bands, the thick-walled, strongly dextrinoid spores that turn dark red in iodine, is strictly and exclusively tied to deciduous trees, specifically birch.
So what were people finding in the conifer forests, those rule-breaking mushrooms we talked about earlier, where people saw a red-banded mushroom growing purely under spruce or pine trees with no birch in sight?
They were entirely different, newly defined species that had evolved a completely different ecological strategy. The 2011 study identified Cortinarius luteornatus, which grows in damp spruce and conifer forests. Visually, it is incredibly similar, but if you look very closely, its veil bands are more pinkish or brick red rather than bright orange.
Subtle differences.
Very subtle. And microscopically, its barcode is different, and its spores are broadly ovoid and indextrinoid. They do not turn dark red when exposed to Melser's iodine.
They also identified Cortinarius paragatus, which also lives in conifer forests, but features a vinaceous wine-red veil.
Yeah.
And Cortinarius pinnigatus, which is highly specific to dry pine heaths, a completely different microhabitat.
Exactly. The bracelet morphology, having that universal veil leave colorful bands on the stem, was a highly successful evolutionary trait shared among a common ancestor.
Right.
But as ancient climates shifted and glaciers advanced and retreated, these fungi were forced to adapt. They underwent host shifting, diverging genetically to occupy different ecological niches and partner with different trees to survive.
And the danger of misidentification doesn't stop within this specific section of sibling species. The forest is full of lookalikes.
It really is.
Right, there are completely different mushrooms way outside the armillatus section that look somewhat similar and can cause serious trouble for an unwary forager. You have Cortinarius bolaris, which also has reddish scales on the stem, but its flesh dramatically bruises yellow or chrome red when you cut it open.
Very alarming to see.
There is Cortinarius vanduzerensis out in the Pacific Northwest, which shares the deep coloration but is incredibly slimy to the touch and has a distinct pointy cap. And then there are bluets, scientifically known as Lepista nuda.
Bluets are a very popular choice edible mushroom. Superficially, to an untrained eye, they can share a similar size, shape, and a brownish-purple hue with some older, weathered Cortinarius species.
But they're different.
Oh, yes. Bluets lack a Cortina entirely. They have no cobweb veil. And if you take a spore print, their spores are a pale pinkish cream, not the dark, rusty brown of a webcap.
Let me push back on this whole taxonomic shakeup, though. Because if you're a listener and you read this 2011 study, you see scientists putting immense effort into splitting one mushroom into 6 different species because of a microscopic DNA tweak, a slight difference in spore shape under a microscope, or the exact species of tree it grows near.
I see where you're going.
To the average person walking in the woods, or even a casual forager looking for dinner, does it really matter if it's armillatus or luteornatus? They look exactly the same.
Sure.
Isn't this just academic pedantry?
If we connect this to the bigger picture, we find that taxonomy is never just academic pedantry. It is quite literally a matter of life and death. The reason scientists obsess over these minute genetic and ecological boundaries is because chemical properties, specifically the ability to synthesize complex, deadly toxins, follow genetic lines.
Okay, that makes sense.
For decades, scientists couldn't accurately test this mushroom for toxins because they were accidentally testing a jumbled mix of 6 different species.
A muddy sample.
Right. By finally using DNA to isolate the true, pure genetic boundaries of Cortinarius armillatus, scientists were able to test the correct organism accurately. And that precise chemical testing is what led them to uncover a hidden, terrifying chemical secret within the true red-banded court.
And this is the massive plot twist of the story. For decades, almost a century, really, if you opened up a standard, respected foraging field guide, it would list the red-banded court under the category of edible or perhaps mediocre but safe.
People were eating it.
People harvested it, people ate it. It wasn't considered a choice culinary delicacy like a morel or a chanterelle, but it went into the frying pan and into the stew.
It did.
Now, because we finally know exactly what it is, we know that it harbors one of the most insidious, terrifying mycotoxins on Earth.
The clarification of its exact identity paved the way for modern targeted chemical analysis. In 2016, a collaborative study led by researcher Dahai Xiao, published in the highly respected journal Toxicon, fundamentally changed everything we knew about the safety of this mushroom in North America.
The methodology of this study was bulletproof. They took fresh specimens from the Harvard forest in Massachusetts.
Yes.
And they didn't just look at the mushrooms and guess the identity. They used the exact lessons from 2011. They sequenced the DNA directly from the birch root tips where the mushrooms were growing to be 100% certain they were analyzing true, genetically verified Cortinarius armillatus.
Once they had the verified tissue, they applied highly advanced analytical chemistry. They utilized high-performance liquid chromatography, or HPLC, coupled with liquid chromatography tandem mass spectrometry, or LC-MSMSS.
That is quite the mouthful.
It is. They use this to screen these mushrooms for specific toxic compounds.
Okay, those acronyms are intimidating. Well, let's provide a really quick explanation of what those machines actually do because they're marvels of modern science.
Go for it.
Imagine these machines as microscopic chemical sorting facilities. They take the raw mushroom tissue, dissolve it into a liquid solvent, and push that liquid under immense pressure through a long, specialized tube. As the liquid travels through the tube, every single molecule is separated based on its specific weight, its size, and its electrical charge. The machine can literally weigh individual molecules as they come out the other end, allowing scientists to find a single, specific toxic needle in a massive biological haystack. They could detect compounds down to the nanogram level.
It is an incredibly precise way to map the chemical makeup of an organism. And when they ran the true Cortinarius armillatus through this chemical sorting facility, the breakthrough was definitive and alarming.
What did they find?
They formally identified Cortinarius armillatus as a novel carrier of Orellanine in North America.
Orellanine. Just the name sounds ominous. What exactly is this compound?
Biochemically, Orellanine is a tetrahydroxylated dienoxidized bipyridine compound.
Again, I'm going to need you to translate that into how it actually affects living tissue.
Think of it structurally. At a molecular level, the shape and behavior of Orellanine are incredibly similar to the highly toxic synthetic herbicides paraquat and diquat.
Herbicides, like weed killer.
Exactly. Which are used industrially to destroy plant tissue. In the human body, Orellanine acts as a devastatingly potent nephrotoxin.
Nephrotoxin.
Nephro refers to the kidneys. It specifically targets and destroys the delicate epithelial cells within the renal tubules of your kidneys.
And the mechanism of how it poisons you is straight out of a psychological thriller novel.
It is deeply unsettling.
It doesn't strike immediately. If you chop up a mushroom containing Orellanine, cook it in butter, and eat it, you don't get sick that night. You don't throw up the next day. You don't get a fever. It causes what is known as delayed acute renal failure.
The latency period is the most terrifying aspect of this toxin. When the Orellanine enters your bloodstream, it specifically accumulates in the kidneys.
Okay.
Once there, it begins to systematically inhibit protein synthesis and RNA polymerase within the kidney cells. It generates massive amounts of oxidative stress-free radicals that literally tear the cellular machinery apart.
Just shredding the cells.
But it does this slowly. The cells are effectively starving and breaking down one by one. Because the damage is cumulative at the microscopic level, outward physical symptoms, extreme unquenchable thirst, severe lower back pain, a sudden decrease in urine output might not appear for 14 to 20 days after ingestion.
20 days. Think about that timeline.
It's a huge gap.
Let's say you go foraging on September 1st. You eat a mixed mushroom stew. You feel completely fine for weeks. You go to work. You live your life.
Right.
Then, on September 20th, your kidneys begin to shut down, and you rush to the emergency room. By the time you were sitting in the ER with end-stage renal disease, facing a lifetime of dialysis or the need for an organ transplant, the mushroom you ate 3 weeks ago is a completely distant memory.
You wouldn't even think of it.
It wouldn't even cross your mind to mention it to the doctor.
Exactly. It makes it incredibly difficult for emergency room physicians and toxicologists to trace the cause of the kidney failure back to the source. It presents identically to other forms of sudden renal failure.
Now, context is highly important here, and we need to be clear about what the Toxicon study actually found. The study didn't claim that the red-banded court is the single most poisonous mushroom in the woods.
No, yeah.
They compared the concentrations of Orellanine in our mushroom to the truly lethal species in the genus, like Cortinarius rubellus, which is aptly named the deadly webcap.
Right. Cortinarius rubellus is a known documented killer in Europe and North America. It contains a truly massive concentration of Orellanine. The researchers measured it at around 26,000 micrograms per gram of dried mushroom tissue.
26,000.
That means roughly 2.6% of the mushroom's total dry weight is made up purely of this kidney-destroying poison.
In stark contrast, the true Cortinarius armillatus specimens from the Harvard forest contain an average of 145 micrograms per gram, so comparatively it's a tiny fraction. It's less than 1% of the concentration found in the deadly webcap.
But 145 micrograms is decidedly not zero, and the danger is compounded by another chemical trait. Alongside the Orellanine, researchers also noted that armillatus acts as a heavy metal accumulator.
Oh, wow.
Its mycelial network acts like an incredibly efficient sponge, actively pulling toxic heavy metals, specifically lead and cadmium, out of the forest soil and concentrating them directly in the flesh of the fruiting body.
Here's where it gets really interesting and a bit terrifying. Eating this mushroom is essentially like playing chemical Russian roulette.
It really is.
Sure, 145 micrograms is considered a sublethal dose. A perfectly healthy, robust adult with kidneys operating at a 100% efficiency might process that minute amount of Orellanine, filter out the cadmium, and never consciously know they ingested a potent toxin.
Which entirely explains why older field guides comfortably called it edible for decades.
But consider the variable human element. If someone has a pre-existing kidney issue, maybe they're older, maybe they're diabetic, maybe they have mild, entirely undiagnosed renal stress, eating this mushroom is essentially kicking their kidneys while they're already down.
That's a great point.
It could be the chemical straw that breaks the camel's back.
That is the crucial clinical perspective. Furthermore, we must remember that 145 micrograms per gram is merely an average derived from a specific batch of mushrooms in a specific Massachusetts forest during a specific year.
Oh, right. It varies.
Natural toxin levels in wild fungi are not standardized like pharmaceuticals. They fluctuate wildly based on the genetic strain of the mushroom, the specific mineral chemistry of the local soil, the weather patterns of that season, and the age of the mushroom.
So it could be much higher.
A specific mushroom you pick off a log in Michigan could theoretically have a significantly higher concentration of toxin than the average found in Harvard. The delayed onset of Orellanine makes it the perfect, untraceable poison in the natural world.
Scary stuff.
Because of these cumulative risks, the modern consensus among mycologists and toxicologists is absolute. All major reputable field guides have officially reclassified Cortinarius armillatus from edible to strictly inedible or poisonous.
So it is permanently off the dinner menu, but just because we can't eat it doesn't mean it doesn't have a profound use for human beings.
True.
In fact, human ingenuity has found a stunning, entirely safe, and historically rich way to utilize this mushroom's incredibly complex chemistry. We've established you absolutely shouldn't put it in your mouth. But textile artists, weavers, and historical dyers absolutely cherish it.
We're moving now from the dark warnings of modern toxicology into the vibrant, creative world of ethnobotany.
The chromatic universe.
Yes, the chromatic universe of natural mushroom dyeing. It turns out the vast Cortinariaceae family is absolutely packed with what botanists call secondary metabolites.
Let's explain secondary metabolites for a second. Primary metabolites are the chemical compounds an organism produces that are strictly necessary for its basic day-to-day survival. things like amino acids for growth or sugars for cellular respiration.
Right, the basics.
Secondary metabolites are extra compounds the organism produces. They aren't required to keep the cells alive, but they give the organism some sort of evolutionary edge in a hostile environment.
A tactical advantage.
Exactly. These might be chemical defenses against grazing insects, compounds that provide UV protection from the sun, or antibacterial agents to fight off soil pathogens.
And in the specific case of the Cortinarius genus, many of these secondary metabolites are a class of chemical compounds known as anthraquinones.
Anthraquinones.
Anthraquinones are incredibly stable, complex molecules that act as highly effective, brilliantly colored natural pigments. And crucially for human textile artists, the chemical structure of these fungal pigments allows them to bond beautifully and permanently with protein-based natural fibers.
The distinction of the fiber is paramount here. When we say protein-based fibers, we're talking about materials derived from animals. Sheep's wool, alpaca fleece, or silk woven from silkworms.
Right.
These fibers are made of long chains of amino acids. By contrast, cellulose fibers like cotton, hemp, or linen come from plants and are made of glucose chains.
A very different structure.
The fungal anthraquinones do not naturally want to bond to plant cellulose. Trying to dye cotton with mushrooms requires a much more complex, multi-step chemical process involving tannins to force the color to stick. But if you have a raw skein of sheep's wool, you are perfectly set up for natural alchemy.
The extraction process itself is remarkably simple, standing in stark contrast to the highly complex chemistry actually occurring inside the dye pot.
How does it work?
Dyers typically take the mushrooms they can use freshly foraged specimens or mushrooms they dehydrated from a previous autumn season. They crumble or chop them up to maximize the surface area exposed to the water.
Okay.
They then simmer these crumbled mushrooms in a pot of hot water. A standard starting recipe aims for a 1-to-1 ratio by weight. 100 grams of dried mushrooms for every 100 grams of wool fiber.
You simmer it down, creating this dark, earthy tea. But if you just toss the wool in that water, the color might wash out the next time it rains.
Right, it won't hold.
The real magic, the true science of natural dyeing that makes the color permanent, relies on the introduction of something called a mordant.
A mordant is typically a naturally occurring metal salt. From a chemical perspective, its job is to act as a molecular binder.
The binder.
It creates a permanent chemical bridge, a coordination complex between the anthraquinone dye pigment floating in the water and the amino acid chains of the wool fiber. It locks the color in, ensuring the resulting dye is wash-fast and light-fast, meaning it won't rapidly fade in the sun.
That's fascinating.
But mordants do more than just bind the dye. Because they are reactive metals, they physically alter the color profile that is extracted from the mushroom. Different metals react with the anthraquinones to produce entirely different visible hues.
Let's break down the exact color outcomes you can get from the red-banded court, because it reads like a magic trick.
You really do.
You have this single earthy brown mushroom. If you use no mordant at all, if you just simmer the mushroom water and steep the untreated wool, you get a very soft, subtle pink or pale beige color. It's nice, but it's muted.
However, if you pre-treat that exact same skein of wool with a tin mordant, specifically a compound called stannous chloride, and drop it into the exact same brown mushroom dye bath, the chemical reaction will instantly turn the wool a vibrant, sunny yellow.
That's crazy. And if you change the metal and use a copper mordant, the chemical bridge alters the light reflection and the wool turns a beautiful earthy green.
And if you use an iron mordant, specifically ferrous sulfate, the reaction darkens the pigment significantly, yielding a deep, rich olivic tone, a dark olive green.
Pink, bright yellow, green, and dark olive, all pulled from the exact same brown mushroom that happens to have red bracelets.
Just by changing the metal.
And the dyers don't stop there, do they?
No.
They can push the chemistry even further by manipulating the pH of the dye bath. If they add a splash of white vinegar to increase the acidity of the water, it shifts the chemical bonds and pulls the hues in one direction. If they add washing soda or soda ash to increase the alkalinity up to around pH 9, it drastically shifts the resulting hues toward the redder or more violet spectrums. It is literally painting with chemistry.
And there is a fascinating, highly specific detail regarding Cortinarius armillatus within the passionate community of natural dyers.
What's that?
Remember those vivid orange-red bracelets on the stem? The remnants of the universal veil.
Yeah, the jewelry.
It turns out that specific tissue contains the absolute highest, most potent concentration of anthracnone pigment in the entire organism.
Oh, wow.
Some dedicated dyers will meticulously sit down with a scalpel and harvest just those tiny red bands from hundreds of mushrooms to create a concentrated dye bath. They're aiming to achieve the most saturated, vibrant reds and pinks possible without the muddying effect of the brown cap tissue.
That is immense dedication to the craft, slicing microscopic bracelets off a mushroom.
It takes a lot of patience.
And the red-banded court is just one member of this incredibly colorful family. The Cortinarius genus is practically a holy grail for natural dyers. You have Cortinarius smithia, which is known as the red-capped court. It yields incredible, deep, true reds and purples that historically rivaled incredibly expensive imported dyes.
Beautiful colors.
You have Cortinarius semisanguineus, the surprise webcap, which looks like a dull, boring brown mushroom on top, but hides vibrant blood-red gills underneath. It yields some of the most vibrant, color-fast oranges and reds in the natural world.
And then, perhaps my favorite, there is the trickster of the family.
The trickster.
Cortinarius violaceus, the violet webcap. It is a visually stunning, deeply saturated purple mushroom that looks like it belongs in a fantasy novel.
A purple mushroom.
But if a novice dyer tries to extract that purple to dye a scarf, they will be deeply disappointed. The specific pigment molecule that makes it purple simply will not bond to the wool fiber. However, the mushroom is so naturally high in accumulated iron from the soil that the liquid dye bath itself turns a dark, inky purple, even if it cannot be used as a traditional textile dye.
So what does this all mean? We started this hour by looking at a relatively mundane brownish mushroom sitting quietly on the damp forest floor.
A very unassuming organism.
And now you realize what is actually happening. You have this complex organism actively pulling heavy metals out of the soil. It is chemically mining phosphorus from solid rock, trading complex sugars, and acting as a high-speed nutrient router for birch trees in a massive underground network.
It's working incredibly hard.
It's hiding a terrifying kidney-destroying toxin that operates on a delayed fuse to avoid detection. And simultaneously, it holds the chemical keys to dyeing a silk scarf a vibrant olive green or a bright yellow using ancient metal salts. It truly makes you wonder what else is hiding in plain sight on a totally mundane Sunday afternoon walk through the woods.
This raises an important question, and I think it is the philosophical core of our discussion today. It challenges how we interact with the natural world.
How so?
Well, our human relationship with nature is constantly evolving. For a very long time, human interaction with a wild mushroom was strictly binary, driven by base survival. Can I eat this for calories or will it kill me?
Right, back to the binary X-ray at the beginning.
Exactly. But an organism is never just one thing. Its inherent value isn't purely culinary, dictated by whether we can put it on a plate. Its true value lies in the immense, multifaceted chemistry it has evolved over millions of years to survive, communicate, and thrive in its specific, highly competitive ecological niche.
Which brings us to the end of our deep dive. Let's distill exactly what we've unpacked today for you, the listener, so you can take this knowledge with you on your next hike. The red-banded court, Cortinarius armillatus, is a masterclass in fungal complexity and disguise.
It requires strict, modern taxonomic scrutiny, specifically DNA sequencing of its ITS barcoding region, to accurately identify it and separate it from a host of genetic sibling imposters that share its environment.
It is an absolutely essential pillar of northern birch forests, acting as the extended mineral mining network of the reach system.
It harbors a low-dose but incredibly insidious delayed-action toxin known as Orellanine.
And it is a sustainable, chromatic treasure trove of anthraquinones for textile artists willing to unlock its chemistry.
A truly multifaceted organism.
Before we sign off, we want to leave you with one final, truly mind-bending thought to mull over, and this comes directly from the cutting edge of the toxicological research we reviewed.
This is fascinating.
We spent a whole segment discussing how terrifying the toxin Orellanine is. We detailed how it acts as a perfect poison, specifically because it is so incredibly targeted. It bypasses the liver, the heart, and the lungs, seeking out and destroying only the renal epithelial tissue of the kidneys, while leaving other organs largely alone.
It is a microscopic guided missile.
But that exact, devastating, highly specific targeting capability is currently catching the eye of modern medical researchers.
Yes, they are actively studying Orellanine's targeted destructive mechanism, not as a poison, but as a potential, highly localized treatment for advanced renal cancer.
Think about the profound, beautiful irony of that for a second. The exact chemical mechanism that makes this humble brown mushroom a deadly, untraceable poison The very thing that forced us to strike it from the foraging menus might one day be chemically harnessed, modified in a lab, and used as a targeted cure to save the very human organ it naturally evolved to destroy.
It is the ultimate testament to the dual nature of these complex chemical compounds. As Paracelsus noted centuries ago, the difference between medicine and poison is often simply a matter of dosage, application, and understanding.
We started this deep dive talking about the comfort of an X-ray, the clean, binary, black and white line of a broken bone. We want the world to be simple. But honestly, after looking at the red-banded court, I think I prefer the muddy waters.
The complexity is what makes it beautiful.
The more you look into the murky, hidden subterranean world of fungi, the more you realize that the blurriness isn't a lack of focus on our part. It is just a staggering depth of biological complexity that we are only barely beginning to understand. Thank you for joining us on this deep dive into the woods. Keep your curiosity alive, watch where you step, and we promise you will never look at a mundane brown mushroom in the forest the same way again.